305. Hello, everyone, and welcome to today's lecture. We're going to be discussing resistance to infection, basically talking about B cells and T cells. Inshallah, it's going to be an easy lecture. And if you have any questions, let me know. You can stop me anytime. So this is my number. If you had any questions throughout the lecture or after the lecture, you can contact me about this lecture or any other lecture. It doesn't matter. Inshallah, I'll be there to help you. Okay, so today's lecture is going to be easy, and let's start with it. Okay, so general introduction. First of all, introduction to the immune system. Now, this is not very important, but it helps you build up a basis to what we were going to talk about now. Now, the immune system, as you all know, it's in your body to fight off the infections. There's two types of immune system. You have the adaptive immune system, and then you have the innate immune system. What is the adaptive immune system? It's basically composed of two cells, lymphocytes. You could have B cells or T cells. And anything that's not lymphocytes falls under the innate immune system. So it could be neutrophils, could be macrophage, could be dendritic cells, or could be the natural killer cells. So anything that's not lymphocytes, it falls under the innate immune system. Okay. Now, when it comes to the lymphocytes, we could have the B cells. B cells act through a process referred to as humoral immunity, which is basically releasing antibodies. Or you could have the T cells. T cells act through cellular immunity, which is basically like the T cells, the killer T cells, which will go into the cell and destroy the cells. Okay. Now, some general differences between the innate and the adaptive immune response. Like in a nutshell, the innate immune system is the system that's found in your body upon birth, and it fights all pathogens, and it's referred to as minimal specificity. What does that mean? It means if you had a bacterial infection, a viral, parasitic, or whatever type of infection, it fights the same way. It takes it, phagocytosis, destroys it. Okay? And it's a rapid response. It occurs within minutes. On the other hand, the adaptive immune response is going to be more specific. So if you had a viral infection on one hand, it's going to be attacked in a certain way that's different than the way that you're going to attack a bacterial infection. Okay. And another important point is that it requires stimulation. So it needs an antigen to bind on it. And we'll discuss that in a second in order to activate it. And since it requires stimulation, it takes more time to activate. And it forms memory so that later activation will be stronger. Okay. So this is just general idea. Now, another thing that we need to discuss before we go into the lecture is major histocompatibility complexes, or the HLA. Now, we'll talk about them more when we talk about T cells, because we start with B cells, but I'm going to explain them now because they're really important to understand, and they're part of the B cells as well. Now, what is a major histocompatibility complex? It's some type of receptor that is going to be placed on top of the cell. And what it does is that it takes this antigen and puts it on top of this cell. Okay? So we have two major histocompatibility complexes, MHC1 and MHC2. MHC1 is going to be found on all nucleated cells. All cells in your body are going to have MHC1. And we said that MHC puts an antigen on top of it, right? So what is this antigen going to be? Some human antigen, some antigen that's found inside your cell. It takes it and puts it on top. Okay, so all your cell or the human cells are going to have MHC1 with that specific antigen. On the other hand, we have an MHC2. It is going to be found on antigen-presenting cells, specifically the professional antigen-presenting cells, but mainly for now, understand that it's antigen-presenting cells. Okay, so antigen-presenting cells. What are these antigen-presenting cells? B cells, macrophages, and dendrites. It's important to know the three types of cells that have MHC2. Okay, so if I said, if any question came to you about which cell has an MHC2 receptor, then it could be it has to be one of these. Okay, I think that's all for the introduction. The last part is the lymphopoiesis. Basically, since we're talking about the adaptive immune response, B cells and T cells, which are lymphocytes, how are they going to be formed in general? They start in the bone marrow. So in the bone marrow, what you get is going to be this hematopoietic stem cells. It could either stay in the bone marrow, where it could become a B cell, or it could go to the thymus, where it could become a T cell. And then once you get your B cell and your T cell, both of them will go to secondary lymphoid organs, such as the spleen and the lymph nodes, where they could do their actions. And in the secondary lymphoid organs, that's where they're going to be activated. This is basically all the general idea. And now we can start with the first lecture, B lymphocytes. All right. So B cells, the first thing we're going to be discussing is the development of B cells. Now, the most important thing when we're talking about stages of development of B cells, it's a little different from T cells. In B cells, what we focus on is what are the changes that are occurring on the B cell receptor. What is the B cell receptor? It's this thing on top. Why is it really important? Because if you go back here, the final function of B cells is that they are going to release antibodies to destroy like become plasma cells to release antibodies to destroy whatever infection they're trying to attack. These antibodies are the B cell receptors. Okay, so when we're discussing the development of B cells, we're discussing the development of these B cell receptors. As you can notice, for example, in the first stage, it's a pro-lymphocyte. What happens here? It's a change in the B cell receptor. In the pre-lymphocyte, there's another change. In the immature lymphocyte, there's a third change. Now we have the IgMs and IgDs. Now, once you have an IgD, IgD is an indication or indicator that this cell has now matured. Okay, since it matured, it can no longer, it's allowed to leave the bone marrow and go to the secondary lymphoid organs. That's why I drew this line, so that after it's mature, it has the IgDs, it has the IgM, it could leave to the secondary lymphoid organs where it could be activated. Now we'll discuss this in details, but this is the general idea. It all depends on the changes that are going to occur on the antibodies. In the first stage, we form the heavy chain, and then we form the light chain, and then it's going to be immature and could be released. All right? Now, the general structure of B cell receptor. No, okay. So the B cell receptor is made of two chains. There is one long chain referred to as the heavy chain, and then there's this small chain referred to as the light chain. Okay. So two important parts in this chain that you need to differentiate between: the variable region and the constant region. The difference between them, like from its name, the variable region, it varies, or like it's different between every B cell, right? So what is the significance of this? When you are creating this B cell, you want to put a B cell receptor or like an antibody on top of it that is going to be different from the antibody that the other B cell is putting. Why is that? Because whenever you're like, you don't know what, in fact, like the B cells, when they're forming, they don't know what the infections that they're going to get, what is the antigen that they're going to have. So you want to have a wide variety or a wide ranges of antibodies to attack as many antigens as you can. Like a different way to think about it, for example, like a small idea to think about it. Think of a person. Okay, I give you a key, and there's 5 doors in front of you, and then this key I tell you that it opens one of these doors. So you don't specifically know which door it opens, but you're gonna try until you find a door that it opens, right? However, if I give you a key that doesn't open any of these doors, or like I gave you a key that opens only one door, it doesn't open any of the other doors. I mean, like if all the keys I gave you open like only one door, then the other 4 doors, you can't open them. That's why we need a variation to open as many antigens as we can. Okay, so this is the variable region. It has the antigen binding site, and it's different between every B cell. The constant region, on the other hand, it's not different. It's different between every antibody, but it's not as variable as the antigen binding region, because basically it only differs on the subtype that you're going to have. So there's only 5 different subtypes you can have for the constant region: the IgMs, IgGs, IgAs, IgEs, or IgDs. We'll discuss them later, okay? But what I need you to know, that the variable region determines the specificity, or like what is the antigen that we're going to bind onto, and the constant region determines the subtype of the antibody, okay? And the last part is that the part of the antigen that you bind onto is called an epitope. Okay, so an antibody binds on the epitope of an antigen. Okay, clear till now, before we go to the development of the B cells? Yeah, this all was a general introduction. Okay. Now, development of B cells. How do we get a different B cell? Like, how do we get a different antibody receptor for every different antigen that we're going to have? It all occurs through a process of DNA genetic rearrangement. What exactly happens here? So again, this is here just, for example, take the idea of a key. This is the key, and then this is the 4 doors. If all of them were the same as the first one, they're not going to open it. That's why we have a variation. Now, how does this variation occur? DNA genetic rearrangement, okay? Now, the first thing that we form in the structure of a B cell, or the antibody, I mean, is going to be the heavy chain. And a way to remember it is, if you remember, this heavy chain was a big chain, and it's the one that's attaching on the base of the cell. So if you don't have the base, or if you don't have this general attachment, the light chain cannot attach on anything, because it holds onto the heavy chain. So that's why we need to create the heavy chain first. Now, when we have the heavy chain, it's going to be called a pro-lymphocyte. Okay? So how is it formed? Basically, this is the genetic sequence of the heavy chain. There is a variable region, a VDJ variable region, and then there is a constant region. The constant region doesn't change. As you can see, the constant region stays the same. However, when we're still forming it first, I told you what we care about is creating the variability. So the variability changes are going to occur in the V and D and the J segments. The first thing that happens is that the D and J segments are separated. So they join together. We create a DJ, and then V joins with the DJ. So first D joins J, then V joins DJ, and then the constant region joins with them. This is how it happens. Now, how do we have the variability? Like if it's all joining together, where do we get this variability from? This basic idea. You don't need to know the numbers. It's just for your understanding. In the V region, we have 44 different segments. In the D region, we have 27. In the J, we have 6. So when you first join the D with the J, you could join D9, J5, or you could join D12, J3, or you could join D12, J2. It's different. And then when you join the V again with them, now you could join V44 or V17 with D9, J5, or V26 with this, or V30, D12, J2. And you can just imagine how many different type of combinations you can create. Okay, so this is all done through a process of alternative splicing in the variable region. Okay, alternative splicing means the first time we broke it down here, or in the other one we broke it down here. That's why we got different type of antibodies. And this part is basically the antigen binding site, how we have the different segments. This is the heavy chain. Once you created the heavy chain, now the light chain has a place to hold onto, which is the light chain now can hold onto the heavy chain. And this part is referred to as the pre-lymphocyte. Now, one way to remember between the difference between a pro-lymphocyte and the pre-lymphocyte, because some people might get confused and forget them, because it's really important to know the differences in them. A lot of exam questions might be coming from it. A way to remember is that pro-lymphocyte, a pro is similar to the word prototype. A prototype is like the first model of something. So if we say a prototype, the first model, so the first model is going to have the heavy chain, okay? And then a pre-lymphocyte, so it comes right before we get the lymphocyte, or a mature lymphocyte. A mature lymphocyte has a complete antibody, right? So just before we get this mature lymphocyte, we're going to add on the light chain. Okay, so I hope this way helps you remember pro and pre. Now, the other step, which is in the pre-cell, how do we get the light chain? A difference is between the light chain and the heavy chain, since the light chain, if you remember, it looked smaller. See, the light chain is smaller. The thing is, there is no more D gene. So it's only V and J. As a result, the V joins with the J, and then the C joins with the VJ, and it's the same concept of changing, okay? Alternative splicing occurs here, similarly to the one that occurs in the constant region, in the heavy chain, and that's how you get the variations. Okay, so variations, or the variable region determines the specificity, and then the constant region to determine the subtype. Clear? This is basically the development of B cells. And now we're going to go to action of B cells. Is it clear, guys? All right. Now, B cells. What's happened? We got the IgM, we have the IgD. Now it's all mature, it's all ready. It could leave the bone marrow and goes to the secondary lymphoid organs. What happens in the secondary lymphoid organs? Can you repeat the alternative splicing part? Okay, so we said that yes, it's the same. I think it's the same for the heavy and the light chain. Now we said that every B cell receptor is going to be specific, or every antibody is specific for a certain antigen. When your body doesn't know which antigen it's going to be getting, the safest option for it is to create as many receptors for as many antigen that it could possibly get. How does it make this wide variation, or this wide range of antibodies? Through the alternative splicing. We have a V segment that has 44 genes, and the D segment that has 27, and the J that has 6 segments. First thing we said, we join D with J. So for example, from the 27D, you can choose whichever one. For example, you can have D1 with J1, or D1 with J2. You feel me? You could get D9, J5, D12, J3, D12, J2, and so on, as many as you can. And then this goes the same on with the V gene segment. So you get, for example, D12 and J3, and then you could have V1 joins that, V2 joins that, V3 joins that, and so on. And then you could change, first we start with that, and then we could change that, and change that, and change that. And this is not through alternative splicing. What does that mean? For example, you cut off this part, so you end up with V3. For example, let's say this is V3, and this is V5. So if you cut off on this part, if you splice this part, you have V3. If you splice this part, now you have V5. So alternative splicing leads to this variation, or this variability. Is it clear? It's not with you for the exam, but I think it's important to understand it. No, never mind. I think it is alternative splicing. The details is not really with you, but you just need to understand that alternative splicing creates the variation. Okay? Alternative splicing creates the variation. That's what's important in the exam. Okay. So now we have a mature B cell. Mature B cells, there is two mechanisms of action. So first thing, it's found in the secondary lymphoid organ. There's the antigen comes through. The part of the antigen that binds is referred to as the epitope. Once the epitope binds on the B cell receptor that is still found on the B cell, it's going to activate it. If it activates it, what happens is there is this specific structure on the B cell receptor known as a signal transduction region. It sends a signal down the cell in order to activate the cell. It tells the cell that, oh, this receptor that we have has bind onto a specific epitope. So let's make more of this B cell receptor. Okay. So the signal transduction region tells the cell to make more antibodies that look the same or have the same specific or the same antigen binding site. So this is the signal transduction region. And once that happens, we could get two B cells, either plasma cells. Plasma cells are the active cells which release antibodies. Plasma cells release the antibodies, or you can have memory B cells. Memory B cells which get the longer lifespan, and then if you get infected another time, then the memory B cells be like, okay, we know this infection, let's immediately convert to plasma cells and attack this infection. Okay. Now, so B cells, the way they attack is through antibodies through the plasma cell. Or remember at the first slide when we talked about MHC, we said that professional antigen presenting cells that have MHC2 were 3 cells: dendrites, macrophages, and B cells. So B cells are a professional antigen presenting cells. So they're going to be presenting their antigen. And once they present this antigen, who is going to be activated with this antigen? It is going to be the helper T cells. Now we're going to discuss the helper T cells and the cytotoxic T cells, which one is going to be activated when we talk about T cells. But for now, know that this B cell is going to activate this helper T cell. And once you activate the helper T cell, is going to release cytokines in order to make more B cells or help you in the development of the B cells. And this is the general idea about it. One other thing here that you'll notice is different between the B cells and the T cells is that B cells could bind an antigen that is freely found or like soluble in the blood. However, the T cells, they can only bind antigen that are presented on MHC molecules. Okay. Cytokines and signal transduction region increase the production of antibodies. Signal transduction region. Okay, yes, yeah, that's exactly what happens. Yeah, cytokines are released by the T cell to make more antibodies, and the signal transduction region is basically from the T cell itself to increase the activation. But all T cells they undergo signal transduction region, and this is the part that you talked about, the independent and the dependent. So the independent is through the signal activation region or the signal transduction region by having an antigen bind freely. Okay, so the antigen freely binds, while that dependent T cell pathway, which is going to be through the cytokines. Important to remember, what is the cytokine that causes proliferation of the B cells? It is going to be interleukin-4. And again, we're going to discuss it more when we talk about T cells and helper T cells. But for now, just know that interleukin-4 creates more B cells. Okay, so interleukin-4 from the T cells, and this T cell is going to have interleukin-4 receptor to create more T cells or more antibodies. Clear? Okay, so the next part, we're going to be talking about what happens in B cells after activation. It is basically 3 different concepts. They're very easy and very simple. Clonal expansion is the first one. It happens like almost immediately after the antigen binds on the B cell, on the B cell receptor. And then the other 2, class switching and affinity maturation, occur on a later time. Now, this is basically a general idea here that, as we said, when we release the T cells or when we release the B cells after they matured, they're going to have IgMs on top and IgDs. But IgD, forget about IgD. It's just there to indicate it's matured. It's not really important. Just focus that they have IgM on top. Okay, so they have an IgM from the constant region. How do we get the other types, the IgAs, IgEs? It all occurs through class switching. Okay, and the other point here is that each B cell has one unique antibody specific for only one antigen, depending on the variation that has occurred. Clonal expansion, the first concept. What happens here is that the antigen has bounded onto the specific B cell that is basically like specific for this antigen. And since this one can attack this specific antigen, we're going to create more of it. And this all is referred to as clonal expansion. It means that we created more of the antibody, or this more of the B cell that releases antibody for this specific antigen. The other ones, and they are found in the region, but we don't want more of them. So they're going to be like inactive or set aside for whatever other thing they need to do. Okay, so clonal expansion means the antibody or the B cell that binds on the specific antigen is going to be expanding and proliferating and duplicating a lot. Clonal expansion. It's very easy. The other one, isotype switching. Now, what you need to know about isotype switching, what happens, like all you need to know is that there's going to be DNA excision, or like a removal of a specific part of the DNA in the constant region. Remember, we said the constant region determines the subtype, in the constant region of the heavy chain, and this leads to formation of other isotypes. Now, I'm going to explain how exactly it happens, but you don't really need to know these parts, okay? So, in the constant region, if you remember, the entire constant chain, let me go back to this slide. Okay, so the entire constant gene has binded onto the VDJ gene. There was no splicing occurring there. Now, if you expand on the entire constant gene, what exactly are the parts of this constant gene? You have a mu part, a delta, a gamma, alpha, an epsilon, and another alpha part. What does a mu part refers to? A mu part is actually the IgM, and the delta part is IgD. The gamma part is IgG. Alpha part is IgA. So we have the entire constant chain that has IgMs, IgDs, IgG, IgA, and IgE. However, since we didn't splice it, we're just at this part. We're at the IgM part. Okay? So later on, what happens after a while, the class switching occurs by DNA excision or alternative splicing in a specific part. For example, if we cut off this part, now we have an IgA. Okay? And since we remove this part, you can no longer put it back on. So this is an irreversible process. Okay? And this is all what happens in isotype switching. Whatever you cut, if you cut here, you get IgD. If you cut here, you get IgG. So isotype switching is basically splicing or DNA excision in the constant region, because the constant region determines the type, and the heavy chain, because the heavy chain, is the one that's going to be attached all the way down. The light chain doesn't really interact with much stuff. The light chain is just sitting there to help you bind stronger on the epitope. But the heavy chain is where all the changes occur. Now, this is the isotype switching. The last part is affinity maturation. Also, very easy. All you need to understand here is that after a while, there is going to be somatic hypermutations. What are somatic hypermutations? They're basically small point mutations in the sequence that basically leads to it having a stronger affinity on binding on the epitope. Affinity maturation. Okay? This is as simple as it could get, and this is all you need to know. Somatic hypermutations, the point mutations in the sequence that causes it to have a stronger affinity on the epitope. All right. Now, before we go to the classes of antibody, do you guys have any questions regarding the development or anything in the process? I'm gonna go over it again real quick, just one last time. So development, there's different stages. There is the pro stage, where we form the heavy chain, and then there is the pre-stage, where we form the light chain, and then there is the immature stage, which has the complete antibody. Okay, and that's going to be released. Antibody has two parts: a variable region and a constant region. The constant region determines the type. The variable region determines the specificity. Okay, now the formation, you understand this part. This part is clear. And then once it's released, it could attack by antibodies, or since it's a professional antigen presenting cell, it could present its antigen to helper T cells, and helper T cells will release cytokines to activate that. Activation could be through either T cell independent pathways or T cell dependent pathways. T cell dependent pathways through the cytokines, or independent pathway directly an antigen to a signal transduction region. Now, the clonal expansion, basically the one that binds, we create more of it. Isotype switching, we cut off DNA part or DNA excision, we remove the part of the DNA through the constant region in the heavy chain to create a different type. And affinity maturation, small mutations in the sequence that cause the antibody to bind on that epitope strongly, or like more, or like in a stronger attachment way. Okay, so we had a question. The mutation is to make it stronger in the attachment, right? Yes, exactly. These mutations just, and it's still binding on the same epitope or the same antigen. It just binds on it stronger, in a stronger way. Okay. Classes of antibodies. This part is important. And you could get questions that ask you, for example, which antibody can cross the placenta, then you need to answer IgG. Now, let's explain everyone. I'll start with the IgMs. IgMs are going to be, remember when we released the mature antibody, the first thing it had was an IgM. So IgMs are the first response or the primary response. We'll discuss that what exactly in a second. But know for now that IgMs are the first response because it is the first antibody that's going to be released. Once there was some splicing that has occurred, now we have an IgG. So it stimulates the secondary response. And you need to know that an IgG response is stronger than an IgM response. Okay, why? Because now, like IgM, when we first created it, it was very general. It was the very basic constant chain or like IgM antibody type. And since now we had created some changes on it, we have an IgG. So IgG is a stronger response. Okay, but what you need to know is that it is the secondary response. And another important point for IgG is that it can cross the placenta. Like in pregnancy, for example, they tell you, like a pregnant woman, which immunoglobulin, or because antibodies could be also referred to as immunoglobulins, IgEs or immunoglobulins. So which antibody could go to the baby is going to be IgG because it can cross the placenta. There is an IgE. You look at the E, it reminds you with the allergies. So IgEs are stimulating allergies. And another important concept is that they're acting against parasites. IgAs are found in any mucosal secretions. So in breast milk, in stomach secretions, in nasopharynx region, in any epithelial region where there is mucus, you're going to have IgAs. And then the last one with IgDs, it's not really important, as I told you. It's just found there to indicate maturation, but you don't really need to care about it anymore. Just focus on these four ones: IgGs, Ms, Es, and A. You really need to know the function of each because a lot of the questions are going to be about a specific, like one-line word to give you the function. Now, this again was here from the lecture. I want to discuss specifically these two parts in IgMs and IgGs. So we said that IgGs, are a stronger response. Why? Because IgGs can act with different ways. They can do neutralization, they can do opsonization, they can do complement fixation. However, IgM, they can only undergo opsonization and complement fixation. IgGs also undergo like other different mechanisms. One thing that I just want to mention here, I don't think you guys need to know what exactly is the mechanism of attack here. And it's mentioned in the slide. That's why I put it, just to tell you, like, you're most likely not going to get asked about, oh, IgG, how does it attack? Or IgM, how does it attack? Mostly you need to understand these different points. Okay. And then again, remember how we said there is a primary or a first response and the second response. So in the primary response, as you can see, we have IgMs. Okay, IgM dominant. In the second response, you have IgGs, IgG dominant. Okay, and you don't really need to focus on the graph. Just know the first response is IgM, second response is IgG. And one last point here is the lag period. So when you first get the infection, there is this period where you're not going to have activation of the lymphocytes, because you remember how we said the adaptive immune system acts later while the innate immune system acts first. So this is referred to as the lag period, like the period between, or like period until there's activation of the adaptive immune response. Here, the main focus is going to be through the innate immune response, right? So how do antibodies cause the damage? Now, also you need to basically take the general idea of each one of the mechanism of attack. So neutralization, what does that mean? It means that the antibody is going to bind on the cell or the toxin to neutralize it. When you're neutralizing it, basically you're making it ineffective, or like it can't cause any harm anymore. So you neutralized it. Okay, so neutralization, you bind on the cells or the toxins. Now, I don't think you guys will get it, but we had a question in our POD exam, because this lecture is really similar to the POD, where it said toxins, what is like the function that is going to be attacking the toxins? It was a neutralization, and we had other options such as opsonization or complement fixation. But what attacks toxins? It was neutralization. Why? Because neutralization, the antibody is going to bind on this toxin, and it's going to be deactivating the toxin. Okay, I just put it for you guys to know in case it comes. The other mechanism is opsonization. What does opsonization means? It basically it's similar to optimization. So if you're optimizing it, you're making this pathogen more suitable for phagocytosis. So if you're opsonizing this pathogen, it's more suitable for phagocytosis by macrophages. This is opsonization. The next mechanism is complement fixation. I think the complement system was discussed in a lecture yesterday, but basically all it does, or like the end product of the complement system, is going to be activating a cell lysis pathway to destroy the cell. So when the antibody binds on this specific, the antibody binds on this cell, for example, consider it a bacteria, it is going to activate the complement system, causing destruction of the bacteria. The other one is agglutination. Agglutination is basically, you're agglutinating them, similar to the blood, similar to the bacteria, similar to whatever pathogen. It's making it slower. You're combining them all together. As a result, they're going to be slower. And the last one is antibody-dependent cell-mediated cytotoxicity. It's a long term, I know, but since it is cell-mediated, all it does is that this antibody is going to bind on a specific pathogen, for example, and then allows the cell that's going to be destroying. What is this cell? It is natural killer cells. Okay, this is it. All you need to know is that antibody binds on a cell and allows natural killer cells to destroy the specific pathogen that we have bound onto. Okay, this is next thing. I'm going to go over clinical correlations, but this is basically all you need to know for B cells. Do you have any questions, guys? Do you want me to go over it again? I'm go over the clinical correlations first, and then if you want me, I'm gonna go over like the entire lecture one last time. I think we have time. Yes, I'm gonna go over like everything one last time. But do you have any questions now that you need me to go back onto? Okay. Clinical correlations, very easy concept you need to understand. The first one is vaccinations and memory cells. What does this mean? Is basically when you start an immune response, or like whenever you do a memory cells that are going to be acting on later, or these vaccinations that help you create memory, the next time you get infected, or the secondary response, it is going to be faster and stronger. Okay, so this is the first infection, it took time, and this is the second infection, it was faster and stronger. So this is all you need to know about this part. Vaccinations and memory cells are going to create a faster and a stronger response. The next one is immunodeficiencies. You could have primary immune deficiencies or secondary immune deficiencies. Primary immune deficiencies are basically intrinsic or genetic changes or genetic deficiencies. Secondary immunodeficiencies due to a secondary cause or due to some cause from outside. So they're going to be environmental causes, such as cancer, treatments. For example, if someone is taking chemotherapy, then chemotherapy lowers the immune system. So that's a secondary immunodeficiency. And the primary immunodeficiency was any type of genetic mutation. I think only thing I don't want to tell you to memorize the example, but just in case, just know BTK is a genetic mutation that is a primary cause. If you had any question that said which, for example, is a primary cause and said treatments, cancers, malnutrition, and you could tell that these are secondary, but BTK, just know BTK is a primary cause. And the last part is hypersensitivity. What is hypersensitivity? Is basically when you say someone has an allergy to something. For example, if you're allergic to peanuts, then you have a hypersensitivity reaction to peanuts. You have different type of hypersensitivity reactions, but just know that allergies are hypersensitivity type 1. Now, type 1 are mediated. Remember when we said allergies, which immunoglobulin was it? It was immunoglobulin type E. And immunoglobulin type E, how do they create this allergic response? By activating mast cells. Mast cells release histamines, and then you're going to get your symptoms. You don't really need to know the symptoms, but histamine, what it does, it causes bronchoconstriction in your lung. That's why when you have asthma, you feel that your lung is collapsing. It causes vasoconstrictions. It causes different stuff. You don't really need to know them. Just know hypersensitivity type 1 or allergies are mediated by immunoglobulins type E when they activate mast cells. And this is all the lecture. Do you guys have any questions? So I know like this lecture, it's really easy. And when I was going on the explanations, I included details that are not really important. So what I did is I made another presentation with basically just the important information that you guys have to memorize. So when we're done, I'm going to go over real quick, and then I'm going to send both to you if you want to study them. So let's go over the question now. The process of VDJ recombination in the heavy chain initiated during which phase of a B cell's development? What do you guys think? Okay. So you guys said B, which is the correct answer, a pro B cell. because ignore this picture. I copied the wrong slide. But since we're talking about heavy chain, it's going to occur at first part, which is like a prototype or a pro B cell. Okay. Next question. What is the name of the process whereby a B cell can produce different isotypes of immunoglobulins with the same specificity? All right, I got the answers. You guys said D, which is correct, class switching. And okay, we have one more question. Which immunoglobulin or antibody, immunoglobulins are the same as antibodies, is present in colostrum, early milk, and protects the mucosal surface of the intestines? C. Yes, immunoglobulin A. You guys are right. And as you guys can see, and these are as hard as the questions could get. They're all the simple recall questions. So that's why I made another presentation with all these simple facts that you guys need to know. All right, any questions about B cells, or do we go to the T cells now? B cells. So formation, pro, pre, immature to mature, antibodies, G arrangement, the VDJ, and then the DJ, and the function, how do they attack? They're activated by T cells, clonal expansion, clonal class switching, affinity maturation. You just need to know one word for each part. Okay. Let's go over T cells. T cells are even easier than B cells. However, in T cells, there is going to be a tad bit more memorization. But concepts-wise, it's very simple and straightforward concepts. Now, before we go to the T cells, you need to know the difference between the immune response mediated by B cells and T cells. When do we want to have B cells acting, and when are the B cells going to be acting? When are the T cells going to act? B cells are attacking invaders outside of the cell. How do you want to remember this concept? So B cells, what do? They release antibodies. So if they're releasing antibodies, it is going to be attacking something that's outside because it's secreting its antibody outside. However, T cells, as we'll discuss in a second, what they do, or like we said it earlier, that they only bind on antigens that are present on MHC molecules, or like antigens that are present on a cell. So since T cells are going to be only acting by binding on cells, they're going to be attacking invaders inside the cells. Okay, so B cells release antibodies outside, T cells are going to bind on the cell, and then they're attacking invaders inside the cells. And this is important when it comes to viral infections, specifically because viruses, as you guys know, viruses, what they do, they invade the cell, and then they start living in the cell and replicating inside the cell. So they're not outside the cell. You cannot attack them by antibodies. Instead, you need something to attack them intracellularly, which is the T cells. All right. Part 2, T cells. So we have two different types of T cells that we're going to discuss. There's the helper T cells, there is the killer T cells or the cytotoxic T cells, and we'll discuss how they act. It's basically a cellular mediated immune response. Humoral was antibodies, cellular was the T cells. And again, focus here, it's intracellular, while the humoral antibodies was extracellular. Okay. T cells, we have two types of T cells. There is the helper T cells and there is the cytotoxic or the killer T cells. Now, what is special about them? Like, how do we know that this T cell is a helper T cell or a cytotoxic T cell? There is a specific receptor that is going to be found, or let's discuss T cells in general. On the T cell, there is three receptors you need to know. The first one is the actual T cell receptor. Attached to the T cell receptor, there is something referred to as CD3. What does CD stands for? Because if you know what CD stands for, it will help you like understand what CD4s and CD8s are. CD is called a cluster of differentiation. So when we say differentiation, it's like when it's making it more specific. So, you know, when you have a differentiation of your cells, or like your embryological cells, they're going to become more specific cells. You're going to become kidney cells or liver cells. So when you have differentiation, so how do you differentiate this cell from other cells? With the specific CD that's going to be found on it. Okay. So every T cell has a T cell receptor with a CD3. It has a CD8. And if it was a helper T cell, this is where the difference occurs. If it was a helper T cell, you're going to have a CD4. If it was a killer T cell, you're going to have CD8. Okay. So helper T cell is a CD4. What does helper T cell do? It helps regulate the immune response. We mentioned helper T cells in the part of the antibodies. Remember when we said after the B cell is activated, there's two ways to increase the activation, either through T cells or through the region that activates the signal transduction region. So the helper T cells are the one that mediating the T-dependent activation, okay? So the helper T cells. While the cytotoxic T cells or the killer T cells, yeah, from their name, they're killing invaders, which are found intracellularly, which is very important to know that they're found intracellularly. And another point is that the B cell receptors were the antibodies, so they're going to be secreted to attack, while the T cell receptors, they're found on the specific cell and they do not leave the cell, okay? Now, CD4 positive and CD8 positives, or like CD4 and CD8 that are found on the T cells, they need to bind on an antigen, right? So how do they get this antigen? Remember, we said antigen is present on an MHC molecule. So MHC molecules are holding antigens and presenting these antigens. So you have two types, MHC2 and MHC1. MHC2 on a specific cell, or like an antigen presenting cell, is going to bind, okay, MHC2 on this antigen presenting cell is going to bind on the CD4. And a way to remember it, I think you guys know this one because the doctor mentioned it, but no, 2 times 4, CD2 times 4 equal 8, and then 1 times 8 equal 8. That's how you know CD8 binds MHC1, while CD4 binds MHC2. But you need to know that CD4 is helper T cell. Now, one way to differentiate that CD8s and MHC1s are for the killer T cells, because like 8 is a bigger number, so it's stronger. And then also the 1, so like it only needs one cell to go destroy go destroy the infection. But CD4 is a smaller number, so it needs MHC2 to like more cells. Now, you try to make some relations in your head to help you memorize them better. Okay. Now, okay, so I think this is basically what we're going to talk about. So a CD8 of the T cell binds on which receptor? It binds on MHC1, while CD4 binds on MHC2. Now, you could find it in other places in your lecture or somewhere else in your questions. You could get HLA, but I don't think you're going to get HLA. But HLA is the same as major histocompatibility complex, or the MHC. Okay, it's a different term for it. All right. Now, the same slide again. This time, we're going to go to the thymus. And I think you guys know from your anatomy, the thymus is a structure that's going to be found in your superior mediastinum in this region. And as you age, it's going to become convoluted. It's like going to shrink and become more fatty until it like almost disappears. Okay, so the thymus, over time, it disappears. All right. Now, development of T cells, it's different from B cells. B cells, we said we focus on antibodies. This time, we're going to focus on the cell and the actual like T cell, how is it going to be changing? Okay. Now, the 3 changes or the 3 steps are going to be maturation, and then positive selection and negative selection. You can consider the thymus as a school for the maturation of the T cells. All right. The first thing, when it first lymphoid progenitor cell, this specific cell goes into the thymus, it is going to be taking on a form known as a double negative. So the first thing is going to become a double negative. How do we say if it's double negative or double positive or like single positive? It all depends on the CD4 or the CD8. Okay, so it starts as a double negative, and then it becomes a double positive, and then it becomes a single positive. Okay, so the third part, it becomes a single positive. Single positive means it has either CD4 or CD8. A double positive means it has CD4 and CD8, so it is doubled on them both. Okay, and then we go through positive selection. It's important to note that positive selection happens in the cortex, negative selection happens in the medulla. Let's discuss them now in more details. All right, so the first thing we said, the common lymphoid progenitors, they're going to go to the thymus. When they first arrive, they're going to be double negatives. So they're going to form a T cell receptor. So they, there is this cell, there's nothing on it. So we're going to form the T cell receptor. Now, T cell receptor on the T cell receptor is not as important as a B cell receptor. There is an alpha chain, there is a beta chain, but there's no any like genetic rearrangements that you need to focus on. Just know that there is 2 chains, alpha chain and the beta chain. Alpha chain is similar to the light chain on the B cells, while the beta chain is similar to the heavy chain on the B cells. And then there is CD3. Remember, we said beside the T cell receptor, always there is a CD3 molecule. And CD3 with the T cells, now we formed a complete T cell receptor. After that, you get the double positive stage. Double positive means it's expressing CD4 and CD8. And then the later stage will be a single positive. How do we choose whether we're going to have a CD4 cell or a CD8 cell? It all depends on whichever MHC it is going to bind onto stronger. Okay, this is going to be moved on to, like, we're discussing it more when we talk about the positive and negative selection. But basically, we said that CD4s bind MHC2, CD8 binds MHC1. So when the cell is still in the thymus and it's still developing, if it binds on an MHC2 stronger, then it becomes CD4, and then the CD8 is gone. If it binds on MHC1 stronger, then it becomes CD8 and the CD4 is gone. Okay, so whichever it binds onto stronger is the one or the form that it will take on. So the single positive, double positive, and double negative. Now, focus or think of the thymus as a school. And in this school, you have macrophages, or the thymic cells, cortical epithelial cells of the thymus, or the epithelial cells, which are going to be presenting the developing thymocytes, or the T cell. They're going to be basically presenting it with the antigen on an MHC1 or MHC2. All right, so the first step is going to be positive selection. It occurs in the cortex, which is the outer layer. So the cortex comes outside, and inside we have the medulla. So in the cortex, we have a process of positive selection. What do we mean by positive selection? Think of positive as basically keeping it alive. Okay? Or like, no, positive is like wants it to bind. Okay, you want to have positive binding. So here, the specific process that happens is that this T cell is presented with an MHC1 and an MHC2. If it binds on the MHC1 or the MHC2, then it will stay alive because there is positive binding, positive to binding. If it doesn't bind, then it's going to die. And how does it die? By apoptosis. Okay, this is positive selection. So positive selection means positive to binding. As a result, so the T cell is going to bind on MHC1 or MHC2. And now remember how we said it becomes single positive, and this part affects how it becomes single positive. If it binds on MHC1 stronger, then it's going to become CD8 positive. Okay, and if it binds on MHC2 stronger, it is going to become MHC1 CD8. If it binds on MHC2 stronger, it is going to become CD4 positive. Okay, so this is positive selection. Negative selection means there is negative binding. Okay, so we don't want it to bind here. Negative selection, we don't want it to bind. It occurs in the medulla. So after it comes from outside the cortex, it's graduated from the cortex. Now it can go to the medulla, to the deeper inner part. So here we have a negative selection process. We don't want it to bind. So what we do is we present this T cell with a self antigen, the antigen that is found in your bodies. So we said all cells, or all human cells, all your cells in your body have a specific antigen. Now, if the T cell is binding on this antigen or on your human antigen, what does that mean? It means that when the T cell goes outside and it's leaked into the bloodstream, it is going to find every cell in your body and thinks that this is a pathogen and start attacking your body. Okay, so as a result, if it does bind on the human body cells or the self antigen, then it's going to die. And again, how does it die? By apoptosis. And if it doesn't bind, means it binds MHC1 and MHC2, but it doesn't bind the ones of the human body. That's why it's a good T cell, so we can release it. So it's graduated and it's released to the secondary lymphoid organs. Okay, and here only 5% graduate. It's not important to know, but like apoptosis, like a lot of the T cells are going to die. Okay, I have this important like this point or note here. It's not with you. I think like you might touch over it when you discuss a lecture about the transfusion of the blood. But what I'm going to talk about here is that MHC1 and MHC2 are different in each human. Okay, so that's why the first step, like since MHC1 and MHC2 are different, the first thing we need to know is that if this T cell that we're gonna mature, does it bind on our MHC1 or our MHC2? Does it bind on this MHC? One second, guys. I'm sorry. Okay, so to know if it binds on this MHC1 or MHC2. Wait, let me go again. I apologize for that. Okay, so every human has a different MHC1 and MHC2. As the T cell is maturing, we want to know if it binds on the MHC1 and MHC2 that are found in our body or not, because the MHC1 and MHC2 are where we're going to present the antigen. So if it can't bind on them, means whatever we presented with antigen is going to be ineffective. So Okay, so understand this part. So different MHCs in different humans, and that's why we have a positive selection process. And now, one other point here is that how is it important in transplants? Basically, MHC1, we said it's found in all nucleated cells. Okay, and we said your cell or your human cell has a self antigen that is going to be presented on the MHC1. So at this rate, if you transplant a person with an MHC that is different than the MHC that you have, because we said like not every human has a different MHC, but there is different MHCs for a group of humans. For example, my MHC might be the same as one of yours, but different from the other person. Okay, and this is important when it comes to transplant. You want to transplant someone with my MHC to his MHC that are going to be identical, because this MHC puts on a specific antigen. If it was different, then what happens is that your T cell thinks that this is an infection and it's going to be attacking it. This part is not important with you, but yeah, I just put it here so that you can know about like why do we have this positive selection and negative selection. Okay, this is basically all about the development of T cells. I'm gonna go over it one last time. We have a maturation, a double negative where there is nothing. We form a T cell receptor, which is gonna have 2 chains and a CD3, and then it becomes a double positive and then a single positive. And this is through positive selection. Positive selection, it binds on MHC or doesn't bind. Negative selection, we kill it if it binds on our antigen, and then it's a complete T cell and it's going to be released. Clear? Am I going too fast, guys? Or is it clear? Okay. Now, T cell receptor, as we said, it's going to be having 2 chains, and these 2 chains, I copied these notes from the slide, but it's not really as important. It has a different chains, has a variable region, it has a constant region. But what you need to know, most importantly, is that CD3, what happens is that when the T cell receptor binds on the antigen, you need a CD3 to activate the cellular signaling. So remember how, like in a B cell, we said there is a signal transduction region. This is the function of the CD3. It's a signal transduction. Therefore, if we have no CD3, the antigen is going to bind on the receptor, but there is going to be no signal transduction. Okay, so there's nothing is going to happen. So a T cell receptor binds on the antigen, a CD3 activates it. Antigen presentation, we discussed it again, but I'm gonna go over it one last time quickly. So it's done on MHC classes. MHC class 1 is found on all nucleated cells, and it's going to activate CD8 cells, which are going to kill intracellular viruses. Okay, now there's one point here that these intracellular viruses, how do they get inside the cell? By basically avoiding phagocytosis. Okay, so they avoid phagocytosis so they could get inside the cell, because I found it in one of the previous questions, so I mentioned it here. Also, it's found in the lecture. Okay, so they avoid phagocytosis. So since they avoided phagocytosis, which is destruction by phagocytes, means they are alive inside the cell. MHC class 2 is found on the professional antigen presenting cells, these 3 cells, macrophages, dendrites, and B cells, to activate CD4 positive. 2 times 4 is 8, and they are going to be controlling the immune response. They do not attack, like they do not mediate any attacks. Attacks are by the CD8 positive. They only control the immune response. Okay, T cell activation. This part is important and it's easy. All you need to know is that for activating T cells, we need an antigen to bind onto it, right? However, it requires 2 signals. The first signal, which is basically the receptor binding on the antigen. So it is going to be the T cell receptor binding on the antigen, and then this antigen is going to be present on MHC2. So you considering, okay, so it's the same for cytotoxic T cells and helper T cells. However, I just put as an example of a helper T cell so that it would make it easier. But if it was cytotoxic T cell, then this would be CD8 and this would be MHC1. But it's the same thing for both, okay? So the worst thing we need to do is bind the antigen through MHC2 and CD4 and T cell receptor with the antigen, okay? And then activate CD3 and whatsoever. Now, the secondary signal, and this is where the change happens, is basically referred to as a co-stimulation signal. There is a CD28 that's going to be binding on B7. Now, CD28, I put this table here so that it could make it easier for you guys. CD28 is found on T cells, while the B7 is found on antigen-presenting cells. A way to remember it is that, so we said CD is a cluster of differentiation. So cluster of differentiation means it's differentiating this cell from other. So CD4 is differentiating the helper T cells and cytotoxic T cells, while CD28, just associated that it's differentiating T cells from other cells. Okay, so CD28. And one other way that I don't know, I just think about it that way. 4 times 6 is equal to 28. 4 multiplied 6 is 28. So CD28 is with the same side as the CD4, so it's going to be on the T cells. If that works for you guys, then inshallah will help you remember it. If it doesn't, try to find a way to remember that the CD28 is associated with the T cell. And now CD28, we said it binds on a B7. There is other names for B7. It could be called CD80. It could be called CD86. You need to know these names as they could come in the exam. You could have B7, you could have CD80, or you could have CD86. Okay, but they are the same thing. They're just different names. This is activation. It's easy. Yeah, you need to have 2 signals. It's a little different because there's 2 signals, but it's easy. All you need to remember is that first thing is going to be binding the antigen, and the second thing is going to be the co-stimulation through CD28 and B7. Okay. As a result, there's going to be activation and release of interleukin-2, which allows proliferation and the clonal expansion of the T cells. Okay. So interleukin-2 is going to be released by these T cells in order to create more T cells. Clear? I think this is like the major part for the development. The other parts is going to be the different concepts. I think you mean 4 times 7. Oh yeah, 4 times 7. My bad. Okay, sorry. 4 times 7. I don't know why I'm taking 6. Okay, that's what happens when you study medicine. T cell lymphocytes. Do you guys have any other questions? Like before about the development of T cells? Let me go from the start. Yeah. So, do you have any question? Oh, oops. Positive, negative selection, graduation. Okay. Let's start now. T cells again. Oh, wait, this is okay. So, T lymphocytes, T helper cells, CD4, helping mediate the response, MHC2, CD8, and cytotoxic MHC1. The first thing we're going to talk about now is how cells CD8 positive or the cytotoxic T cells, how do they act? You need to remember 2 things that they're going to be releasing: perforins and the granzymes. What do perforins do? They are going to create the holes inside the cell, and then the granzymes go inside the cell, granzymes go into the cell and activate the apoptosis of the cell. Okay? And as a result, the cell is going to die. That's all you need to know. Cytotoxic T cells secrete porphyrins and granzymes. Perforins are going to be creating the holes, granzymes are going to be activating the apoptosis system, and the cell is going to die. Again, they're going to be binding on the MHC1 to activate or to kill intracellular pathogens. The other one is helper T cell. Okay, this one is tough because it's basically purely memorization, and I think you probably need to know all of them. So I'm sorry about that. So I'll try to make it simpler for you, like a way to remember it. Okay, so helper T cells. We have different type of helper T cells. We have these 4 types. T helper 17, you don't really need to care about it now. That's the first, and the second, and the T regulatory cell. Let's start with T helper 1. T helper 1, what does it do? It activates the cellular response. So activates macrophages and neutrophils. Okay, this is the function of it. Now, how to remember that T helper 1 is going to be activated by interleukin 12 is basically, you see the one here, try to associate this one with this one. So T helper 1 is activated by interleukin 12s. Okay, and what does it release in order to recruit these guys? Interferon gamma. Try also to remember the one is the same as this I. So similar to interferon, IFN is referred to as interferon gamma. Okay, so this is T helper 1. It's pure memorization, but just try finding ways to remember them. Okay, so for example, you could use the 1 and the 12. This is how I remembered it. The second one is T helper cells type 2. T helper cells type 2 are stimulating B cells or the humoral immunity. Remember when we said B cells are going to be, when we said the proliferation of B cells was T cell dependent. Wait, let me go back to this just so that you can link both slides together and then it will make sense. Okay, yes, see this part? So we said that B cells are going to be activated by helper T cells. What specific helper T cell was it? It was helper T cells type 2. Okay, so T helper cells type 2 release interleukins 4 in order to activate the B cells. All right. Now we can go back to the B cells here, or the helper T cells. Okay, so T helper cells type 2 are the one activating the B cells now by releasing interleukin-4. And similar to that, which makes it easier that since they're releasing interleukin-4, and you already know that from the B cell part, they're going to be activated by interleukin-4s as well. So, this helps you remember it immediately that T helper cells type 2 release interleukin-4s and they're activated by interleukin-4s. So it's the same thing. And also they release interleukin-5s and interleukin-13s. Just know these in case they asked you. But I think if it was to be a question about them, it wouldn't be like which one releases interleukin-5. It probably would be which one releases interleukin-4, 5, and 13. But just in case. And then the last one is Treg. Treg means T regulator T cells or helper T cells. What did it do? It regulates the immune response. Specifically, it terminates the immune response. Why is it important? Because if you don't have your T regulatory cells, means any immune response that starts in your body that involves T cells, it won't stop. And your T cells are going to start attacking this cell and the other pathogen. And since you're not able to stop them, they're going to be attacking your own human body. So your T regulatory cells are very important. Now, what do they release? Similar to, okay, never mind. Okay, so interleukin-10s, this is a structured substance that they release. Remember the 0, so 1, 0. The 0 is for terminating, so 0 to terminate the immune response. And TGF, a way to remember it, or like how I remember it, is what does TGF stand for? TGF is referred to as tumor growth factor. So since it has growth, or the TG, growth factor, so G, so just remember growth, which is basically trying to stop the immune attack. Okay, so T regulatory cell is releasing TGF, or tumor growth factor, and interleukin-1, 0, or 10. The 0 is to terminate the immune response. And it's activated by interleukin-2. I don't think there's a way to remember interleukin-2, but just interleukin-2. So maybe 1 is immune response, 2 is no more immune response. So maybe that. Thing is, like when you later take on POD, like I only remembered it because in POD we have interleukin-1s. Interleukin-1s is going to be stimulating immune response. So if interleukin-1 stimulates the immune response, interleukin-2 ends the immune response. But you guys don't have to worry about interleukin-1. Just know that interleukin-2 ends the immune response by activating T regulatory cells. Okay? So I think this is the last slide, but before we go into it, do you have any questions about the T cells? All right, let's go into the last slide. Okay, so here we're going to be discussing basically different infections and what is the immune response that you're going to have from it. If you had an intracellular infection that included viruses, like don't worry about some bacteria, just viruses, okay? So viruses are intracellular infections. If you have intracellular infections, since they're going to be avoiding the phagocytosis or phagodestruction, they're going to be located in the cytosol. And so since they're found in the cytosol, they are going to be presenting their antigens on MHC class 1, and they're going to be destroying them by CD8 positive, similar to the cytotoxic T cells. Okay? Clear? You know this part. The other one was the extracellular infection. If it's an infection that's outside, you're going to have an antigen that is going to be presented on MHC class 2. Basically, so if it's an extracellular infection, your macrophages are going to come. They're going to take in this bacteria and destroy it through the phagosomes. And then they're going to take, since this is a macrophage or a professional antigen presenting cell, where is it going to put the antigen? On MHC class 2. And what helper T cells does it activate? CD4 positive helper T cells type 1. Okay. So since it's an extracellular infection or a bacterial infection, it activates type 1 cells. What does it activate type 1 cells? Because type 1 cells recruit your neutrophils, recruit more macrophages, recruit more cellular mediated immunity to destroy the cells that are found outside. Okay. So this is another way to know, or the second type of infection. And the last one, which is basically extracellular proteins, which include vaccines and toxins. I remember we had this question in our exam, and the answer was T helper cells type 2. And I got it wrong, so don't make my mistake. And just remember that toxins, now how to remember it? Basically, remember what we said, that toxins are going to be neutralized. Okay, so toxins are going to be attacked by neutralization. And this neutralization requires antibodies, right? So this neutralization requires antibodies. So it's going to activate T helper cells type 2. T helper cells type 2 activate the B cell response, okay? And since it's the T helper cell, so it's MHC class 2, I don't think you really need to know endosome, but just know toxins, and then add beside toxins that also vaccines act through the same way. Okay, so toxins require something to neutralize them, which is an antibody, and antibodies are made by B cells, and B cells act by T helper cells type 2. Okay, so this is what you need to know from this table, and this is for the helper T cells. Okay, clinical correlation, this is the last part. When you have an HIV infection, what happens is that you're going to have no more helper T cells. So attacks the helper T cells, there's no more helper T cells. As a result, there is no more activation of the B cells by interleukin-4. There is no more activation of the cellular immunity by activating neutrophils or macrophages by T helper cells type 1. This is it. Helper T cells or HIV affects the helper T cells. Is it clear, guys? Right. You have any questions for B cells or T cells? All right. Do you want to go over the questions? All right. So let's start with the first question. Which of the following cell types is involved in both the humoral and the cell-mediated immune response? Okay, let's solve this question. Then I'm gonna go over your slide again, just because we started this question. Okay. So I got 2 answers for this question. 2 people said E. Does anyone else want to try? And then I'm gonna go over your slide. I'm gonna take one more answer and then I'm gonna say the correct answer. Which of the following activates the humoral response and the cell-mediated response? Okay. Yes, you guys are right. It is E, the helper T cells. And again, why is that? Because we said activates the cellular response by the macrophages and the neutrophils, and the humoral response by the B cells and the antibodies. Okay, I had a question to go back over the T cell maturation slide. So do you mean this slide? Is there a way to clear my ink? Yes. Okay. So okay, so this slide, basically what we're talking about here is when the common lymphoid progenitor cells, they are released from the bone marrow. If it was a B cell, it stays in the bone marrow. If it's a T cell, it goes to the thymus. Now, as it enters the thymus, it's still fresh. There is nothing on the cell. So we refer to it as a double negative. There is nothing on it. When we talk about double negative or double positive, specifically, we're talking about CD4s and CD8s. Okay. Now, when it first enters the thymus, first thing we need to do is we create a T cell receptor and attach it on this cell so that we know that this is a T cell. T cell receptors is made of 2 chains, an alpha and a beta chain. They're not as important as the B cell receptor chains, but there's 2 chains, alpha and beta chain. And beside them, there's another part known as a CD3. CD3, what is the function of CD3? When the T cell receptor binds on an antigen, the CD3 activates the cellular processes inside the T cell so that it can basically create new proteins, or similar to the signal transduction region. So basically CD3 is a signal transduction region. Once you have a T cell receptor on this thymus, the next thing will be there's going to be a CD4 attached to it and the CD8 attached to it. You don't really need to know what happens, but basically there's nothing on it. There's a T cell receptor. Now there's a CD4 and a CD8. Since it has both, it's referred to as a double positive. Okay. Now, from the next slide, you remember we said there's the positive selection process. Once it has both CD4 and CD8, it can now bind on MHC1 and MHC2. If it binds on MHC1 stronger, it's going to create a CD8 positive cell, and the CD4 will get downregulated. If it binds on MHC2 stronger, it is going to have a CD4 positive cell, and the CD8 will get downregulated until there is no more CD8. So after a while, we're going to have upregulation of CD4 if it binds on MHC2 and downregulation of CD8. So the cell becomes a single positive cell with a T cell receptor that has a CD3 and a CD4 cell. Okay. Let's go back to the questions. Do you guys have any other questions about this lecture? Question 2. Fragments of cytoplasmic pathogens which escape phagocytosis are presented on a T cell by which of the following means? What do you guys think? Okay, I got an A. I only got one answer so far. Okay, I got two now. Okay, most of you, or like all of you, said A, placement into endocytic vesicles and complexing with an MHC class 2 molecule. Honestly, I'm not really sure of the answer. One second, let me just check. I think it was D. Let me check. Okay, so I got one D. Yes, the correct answer was D. Okay, so why is it D exactly? Because, second. Okay, if we go back to the previous slide, what we're talking about here is, so we're talking about it escaped phagocytosis, okay? So right now, it is an intracellular pathogen. It's inside the cytoplasm. It's an intracellular pathogen. And intracellular pathogens are similar to viruses, right? So they can no longer be placed on MHC class 2 molecules. MHC class 2 molecules are only presented after phagocytosis. MHC class 1 are presenting intracellular stuff. So anything inside the cell, since they're found in all nucleated cell, that's how you remember. So anything inside the cell is going to be presented on MHC class 1. As a result, it's going to be attacking cytotoxic T cells. And if you remember from this slide, intracellular infections are found in the cytosol. They're going to be affecting MHC class 1 and the CD8 positive cells. Oops. Okay. You guys understand it now? If we said MHC class 2, it would have been everything that has been phagocytosed. So anything that's outside the cell, such as toxins, vaccines, or the bacteria outside the cell. Okay? Antigen presenting cell, again, this was another slide that said pathogens that had invaded phagocytosis are presented on MHC class 1. This is from the B cell lecture. The antibody isotype mainly produced during the primary immune response is IgGMs, IgGs, IgEs, IgAs, and IgD. Okay, you guys got it right. It's IgM because it's the primary immune response. Question 4. Recognition of self versus non-self by the adaptive immune system is accomplished in which of the following ways? Is it B cells or is it T cells? And is it occurring in the thymus or the bursa of Fabricius? D. Okay, you guys said D, which is exposure to our body's antigen in the thymus, which is the correct answer. This is the positive selection part because it's not B cells. B cells formation is just B cell receptor, and I have no clue what bursa of Fabricius is, so it's D. Okay, question 5. A 3-year-old child who has frequent ear infections is found to have reduced immunoglobulin levels and is unresponsive to vaccinations with tetanus toxoid. However, the child has normal skin test reactivity to a common environmental antigen, which cell lineage is not functioning normally. So we have a D, a C, and an A. Okay, so okay, we got 2 C's, 2 D's, I think, and 2 A's. So the correct answer here, as you guys said, is C. And as one of you sent me in chat, it is because it's activation of the helper T cells type 2. Okay, so why exactly here is it not B cells, even though B cells do release antibodies? Because the thing is that there's only reduced immunoglobulin levels. Wait, 1 second. Yes, because it is normal skin test reactivity. Okay, so since it's normal skin test reactivity, I don't think it's going to be mentioned with you. I shouldn't have added this question since it's a little complicated, but what I wanted to focus on was the part that since it's a tetanus toxoid, it's going to be activated by the MHC class 2 cells, okay? Because toxins are activated by MHC class 2. But he isn't being responsive to vaccination. Wait, where? So to have reduced immunoglobulin levels and as unresponsive to vaccination with tetanus toxoid. No, he is unresponsive to vaccination with tetanus toxoid means, okay, that's true. He's not developing the memory. But here it says that there is normal skin test reactivity. Okay, honestly, I'm not sure anymore. You do have a point. I got this question from one of the previous batches. So the answer was helper T cells there. So I'm not really sure. So I'm not really sure, honestly. I have to check this question and look back at it again. So like, okay, forget about this question. Just know the concept that toxins are going to be MHC class 2. If it was to come as a toxin, it would be a more direct question. I'll have to look at that question again. I'll apologize for that. Yeah. Now, I have other questions that I put here, but for the time's sake, I didn't put them in the slide. Do you guys want to go over the other questions as well? Wait, no, the answer is B cells. Wait, okay. I'm gonna go over the question again. I'll fix it in the presentation and then I'll discuss it with you guys again. Okay, I apologize for this. I'll forget about this question. So do you guys want to go over the other questions first? Okay, let's go over them. Okay. Question 6. A blood sample from an individual with systemic lupus erythematosus, projected mapping T cell receptors. Hold up 1 second, let me just fix up my screen. Okay. So many T cells were discovered to express receptors specific for autologous antigens, failure of which receptor in the thymus leads to large numbers of autoreactive T cells in the patient's blood. Those questions, I think I got them from Lippincot's book. So, but they're similar to your concepts. So inshallah, it's going to be easy. Failure of which process in the thymus leads to large numbers of autoreactive T cells in the patient's body? Yes, you guys got it right. The correct answer is D. Why is it D? Because basically negative selection, what it does is ends the autoreactive cells. Okay. Next question. T helper cells interacting with antigen presenting dendritic cells require signals generated by the molecular interactions of the T cell receptor with the MHC peptide complex. Additionally, co-stimulation is required to amplify the initial TCR signal provided through the T cell CD28 molecule interaction with which of the following dendritic cell molecules? So your question here is CD28 interaction with what? There is a D and there's a bunch of A's. I'll give you guys a second if anyone else wants to try. Two D's and three A's. Okay, so the correct answer here is actually it's D, CD80 and CD86. And let's go back to the slide 1 second. This is talking about the CD28, which is multiplied by 7, not by 6. CD28, oh yes. Okay, so this is a way to remember it. Since it's multiplied by 7, it bonds on B7. And then B7 is CD80 and CD86. Now, the actual choice there was CD80. And CD80, as I said, is another way to say B7. Okay. Now, for those that said the answer was, one second, let me go. Okay, so for those that said A, which is CD4, CD4 binds on MHC class 2. Okay, CD4 binds on MHC2. CD8 binds on MHC class 1. So it can't be CD4, it can't be CD8. CD45, we didn't discuss it. CD80, CD86 is another word for B7, and CD28 is multiplied by 7, so it binds on B7. Okay, and then CD152 also we didn't discuss it. That's why the answer is D. Clear? Okay. Next question. Dendritic cells, macrophages, and what other cell types are considered professional antigen-presenting cells capable of antigen presentation on T helper cells or to T helper cells? You guys are right. The answer is A, B cells. Oh, there was an E, neutrophils. It's pure memorization or recall. Okay, so for neutrophils, the thing is, neutrophils, what do? Neutrophils phagocytose, or like take in the organism and destroy it. Okay, so since they're destroying it, they're not really presenting antigens. Mast cells, what do? They release histamines. Eosinophils, I'm pretty sure, like if I'm not mistaken, they were responsible for like parasitic infections. Basophils, I don't really remember. Oh, one thing like for HLS, it's not in this lecture, but I think it's the leukocyte and in the leukocytes lecture for hematology, Dr. Rayhan, you need to know what does the basophils release. I don't really remember the name, but I think they release a specific substance or major acidic or major basic protein. So yeah, remember which one releases major basic protein, as it could be a question that you get. Eosinophils attack parasites and one. Yes. So, or maybe eosinophils was the one that releases the major basic protein. But remember which one releases that, because you could either get an eosinophils releases what to attack, or which of the following releases the major basic protein. Okay, so you guys said B cells, and it's basically the three cells, which is B cells, the macrophages, and the dendrites. Okay. We have two more questions. What is the source for interleukin 4, 5, and 13? Got E, E, E. You guys are right. Most of you said E, which is the correct answer, because whenever you see interleukins, immediately cross out anything else. So it's either going to be T helper 1 and T helper 2. Okay, I want to ask you a question here, a follow-up question. So they present their antigen, these cells, on MHC class 1 or class 2? The professional antigen presenting cells. Class 2. Yes, you're right. Okay, and what cells? Okay, so another question, a follow-up question for this part. What is the interleukin that is released to stimulate B cell, or like to stimulate the formation of T helper cells type 2? Yes, you answered it already. What is the interleukin that is stimulating formation of T helper types 2? Yep, you guys are right. Interleukin 4 is released and it's stimulating it. Okay, no more follow-up questions. And what does it mediate? What is the result of this activation? Activating interleukin 4, 5, and 13 causes humoral immunity. Yep. Okay, this is the last question. If a person had a genetic defect affecting perforin production, which cells and immune function would be affected? And then there's a follow-up question for you guys. What is the function of perforin? If you guys are right, you got the question. It's A. Follow-up question, what is the function of perforins? Yep, makes the holes. You guys are right. Okay, one last follow-up question. What is the substance released by cytotoxic T cells that stimulates apoptosis or the cell death? Coenzymes. I think that's all. I'll check on this question again, but all I wanted, like my main point from that question, was to know that toxin or the tetanus toxoid is going to be activating the CD4 positive T helper type 2 cells. But I'll make sure of the question again and change it in the answer slide. The answer is here. All right. Do you guys have any question about the lecture? Is it clear? Inshallah. All right. One last thing that I just hold up. So, okay, let me present this. So I made another lecture here real quick with basically all the terms that you need to memorize. Basically, I simplified everything, no explanations, just what you need to memorize. So this is the differences between them. This is the MEC class 1, MEC class 2, B cells, development. This is what you need to know from the development. This is what you need to know from the activation. This is the subtypes. This is the function. This is the difference between B cells and T cells. Clinical correlations, the different clinical correlations we had for B cells, and then the T cells. That is the formation, the activation, the structure, and then the different function of the helper T cells, cytotoxic T cells. Okay, and right preference. Okay, so there's preference. Okay, so this is all you need to know for this lecture in general. Okay. Do you guys have any questions? Okay, you're welcome. I'm glad you guys. And so my number also is in the slide. If you had any questions later on and regarding this lecture or regarding any other lecture, I'll be free to help, inshallah. And best of luck for you guys' exam. Inshallah, you all do great. And good luck.